Hello, everybody. My name is Megan Golden, and I am one of the co-founders of Mission Cure and now the CEO. And I am here with Linda Martin, who is also a co-founder, and she is now Mission Cure's board chair. I also am joined by my Mission Cure colleagues, Shruti Mirashankar, Olivia Zaiden, Sarah Svensson, and Isabel Aguirre. Welcome to Mission Cure's webinar on stem cell therapy for pancreatitis patients and families, and also welcome to our community of clinicians and researchers and all of the other stakeholders who are joining us today. We have over 300 people registered for this webinar, which is a record for us. So first, a little bit about Mission Cure. I co-founded Mission Cure with my brother Eric and with Linda in 2017. Both Eric and Linda's daughter Amy had been diagnosed with chronic pancreatitis after many years of pain and misdiagnosis. Doctors said that there was no treatment, no cure, and the only thing that they could do was try to manage the pain through palliative care. That was not good enough, and we launched Mission Cure to change that. We are a bit different from the typical disease foundation. When we launched, one of our first steps was to survey patients to see what outcomes were most important to them. Number one was pain. And then with help from leading pancreas experts, we developed a strategy to improve those outcomes by developing therapies, taking what's called multiple shots on goal. And we are making good progress. Our approach is guided both by the community needs and by the science behind potential treatments. This webinar is part of our patient education program, which is supported by AbbVie. Thank you. So before we begin, I'm excited to announce that we will soon launch a pilot study for an app that pancreatitis patients can use, along with wearable technologies to track their symptoms and what's happening in their bodies. If it works, this technology could give insights into what drives pancreatitis attacks and what happens in the body when there's a flare or an attack. And this can help patients manage their disease and also lay the groundwork for new therapies. If selected to participate, you will be given an Oura Ring, which you see on the slide there, and also an Empatica Embrace Plus wristband, which is kind of like an Apple Watch. And you'll be expected to complete daily tasks that should take around 5 minutes. And you'll be able to contribute to how this technology is created with feedback that you provide throughout this 5-month study. We won't officially begin recruiting participants until February 2025, but in the meantime, feel free to reach out to Daniel with any questions about the app or the study. His email is here on the slide. So now I want to introduce Shruti Mirashankar, who leads our patient education program, to go over our agenda for the day and to introduce our topic of today's webinar. Shruti?
Thank you, Megan. A warm welcome to all our live attendees and people from around the world watching this webinar. Our agenda is outlined on this slide. Today, you'll hear from Dr. Wan about stem cells and the buzz surrounding stem cell therapies for treating diseases. She will also present the stem cell clinical trials that are currently recruiting. Then, we'll hear from our patient speaker, Alice Voss, about her health journey and why she chose to participate in a clinical trial. We organized this webinar because stem cell therapy is one of the most talked about advances in medical science today. For the pancreatitis community, understanding how stem cells might offer new treatment possibilities is exciting and empowering. In this session, we'll cover the basics of stem cells, their potential for treating pancreatitis, and the clinical trials that will help advance these therapies towards FDA approval. Our goal is simple: to inform, encourage, and empower our community. By learning more about stem cell therapies, we hope to help you make informed decisions about your care and inspire hope for better treatment options. Let's dive right in. I'm thrilled to introduce our first guest speaker, Dr. Hoon Jin Won, Professor and Scientific Director of the Center for Cellular Therapy at the Medical University of South Carolina. Dr. Won's research expertise is in stem cell therapy and transplantation immunology, with a special emphasis on treating chronic pancreatitis and diabetes. She is guiding cutting-edge stem cell research, focusing on translating these innovations into real-world therapies. Today, she'll introduce us to the world of stem cells and share her expertise on the potential of stem cells in treating pancreatitis. Dr. Won, over to you.
Good morning. Thanks to Mission Cure for organizing this webinar. It's truly a great honor to have the opportunity to introduce our studies here. My group at the Medical University of South Carolina started as a basic science lab focused on autologous islet transplantation. Over time, we expanded our focus to exploring mesenchymal stem cell therapy. This research enabled us to transition into a new bench to bedside clinical studies for treating chronic pancreatitis and diabetes. The pancreas is an important organ in the digestive systems. It's located in the upper abdomen, just behind the stomach and next to the intestine. It's made of a network of small tubes called pancreatic ducts that release the digestive fluid into the small intestine. The pancreas has two parts that are responsible for its major function, the exocrine and the endocrine compartments. The exocrine compartment, which makes up most of the pancreas, contains acinar cells and duct cells. Its main job is to produce enzymes that help to break down food during digestion. The endocrine compartment, which accounts for only about 2% of the pancreas, and is made of pancreatic beta cells and other specialized cells, produce important hormones like insulin, which help regulate blood glucose levels. Together, the exocrine and the endocrine functions maintain the body's energy balance and overall fuel management. Chronic pancreatitis, or CP, is caused by chronic inflammation of the pancreas, leading to irreversible destructions. The damage could include the death of acinar cells, shrinking of the pancreatic tissue, change in the structure of the pancreatic ducts, and buildup of scar tissue. And CP can impair both the pancreas' ability to produce digestive enzyme, that's the exocrine function, and also hormones like insulin, which related to the endocrine function. One of the most dramatic and debilitating features of CP is persistent abdominal pain, which affects more than 90% of the CP patients with the condition. This pain is often described as the worst pain one can encounter in life and can have a major impact on daily life and overall quality of life of the patients. The primary challenge in treating chronic pancreatitis is managing this severe and often intractable pain, which can be difficult to control with medication. My lab has been exploring effective and novel ways to treat chronic pancreatitis with stem cells. So what are stem cells? Stem cells are unique, unspecified cells that can self-renew, meaning that they can produce more stem cells to maintain a continuous supply. They can also differentiate or transform into specialized cell types that the body needs. This ability makes stem cells essential for repairing and regenerating damaged tissue, helping the body heal and recover. Stem cells are characterized based on their potency. Potency refers to how many different types of cells they can become. This classification helps determine their potential for regeneration and repair. They are totipotent stem cells, like fertilized egg, that can generate all 220 different types of cells in the body. They are pluripotent stem cells, also called PSCs, can generate all the major cell types derived from the 3 embryonic germ layers, but they cannot form actual embryonic tissues. They are multipotent stem cells, like mesenchymal stem cells and hematopoietic stem cells, they can develop into only a limited range of cell types depending on their tissue of origin. While stem cell therapy has been explored in clinical trials, the US FDA only approved hematopoietic stem cells for treating certain blood cancers at the time being. Our research focuses on mesenchymal stem cells, which are the multipotent stem cells, but they return some differentiation properties. They are please note they are distinct from embryonic stem cells, making them a promising option for therapeutic use without the ethical concerns associated with embryonic stem cells. So what are mesenchymal stem cells, or MSCs? They are a rare type of adult stem cells mainly found in connective tissues, such as umbilical cord, bone marrow, fat tissue, among many other tissues. MSCs can differentiate into different specialized cell types, including bone cells, cartilage cells, and fat cells. Because of this versatility, MSCs are of great interest for research and therapies focused on tissue repair and regeneration. Why use MSCs? MSCs, first of all, is relatively easy to isolate. They can grow in large numbers in a laboratory petri dish and still maintain their ability to support therapeutic functions. This makes them promising candidate for use in medical treatments. Most importantly, they have pleiotropic effect. What does that mean? That means they can influence the body in different ways. This may include regulating the immune system, reducing inflammation, preventing or reversing tissue scarring, and promoting the formation of new blood vessel. That's called pre-angiogenesis effect. This effect are hard to achieve with a single drug, which is also make MSC therapy especially valuable for treating diseases with complex symptoms like chronic pancreatitis. So a lot of scientists spend a lot of time studying MSC therapy. We believe that MSC work through several key mechanisms to support healing and repair. These include number one, direct differentiation into specialized cells to replace the damaged cells. And through cell-cell contact, they contact with surrounding cells to support tissue repair. And paracrine effect, MSC release proteins that promote cell growth and healing. MSC also functions through releasing of extracellular vesicles and mitochondria. These tiny particles help deliver healing signals and support damaged cells by improving energy production and other essential functions. There are two major types of stem cell therapy, autologous and allogeneic cell therapy. Autologous cell therapy uses the patient's own cells for treatment, while allogenic cell therapy uses cells from a healthy donor instead of the patient's own cells. Both types of therapy have their own pros and cons. For example, there's no immune rejection related to autologous cell therapy, and no donor search is needed. But patients often have to undergo sample collection process. In contrast, allogenic cell therapy may be recognized and rejected by the recipient, although the cells can be stored and used as an off-the-shelf treatment when needed, which makes the process faster and more convenient for patients. MSCs are flexible because they have very low level expression of certain markers that reduce the risk of immune rejection. We call it immunoprivileged. These markers including HLA class 1, class 2, CD40, 80, and 86. So this means that both autologous and allogenic MSCs can be effective, provided the disease itself doesn't impact the therapeutic property of the cells. This is true for chronic pancreatitis. A postdoc from my lab reported that years ago that chronic pancreatitis does not impact the therapeutic function of MSCs as showed here. MSC therapies has been tested in numerous animal studies and clinical trials for various diseases. This includes diabetes, rheumatoid arthritis, multiple sclerosis, and others. The results from these studies supported the safety, effectiveness, and long-lasting benefit of MSC therapy in treating different conditions. MSC therapy has also shown promising results in reducing chronic pain and inflammation linked with conditions such as knee osteoarthritis, critical limb ischemia, and diabetes neuropathy. In some of these studies, MUSC were found in their spinal cord, so maybe they migrate to the tissue and exert protection. So over the past 10 years, our lab has been studying the therapeutic effect of MSCs in several mouse models that mimic key aspects of chronic pancreatitis. Here an overview of some findings from our research using the TMBS-induced chronic pancreatitis mouse model. We observed that treatment with a chemical called TMBS reduced the pancreatic volume, primarily affecting the exocrine part of the pancreas. Remember, they mix about 98% of the pancreatic tissue. The images here of the pancreas from these mice illustrated this reduction. However, when we treat these mice with human MSCs, we found dramatic improvements. Specifically, MSC treatment reduced the death of acinar cells and improved overall pancreatic volume. Further analysis showed that mice treated with MSCs had better preserved pancreatic structure and reduced the level of fibrosis. I didn't show the fibrosis slides here, but we do have data confirm that. This has been confirmed by scanning the entire pancreas with hematocilin and using staining. This is a lab technology to identify different parts of the cells under a light microscope. And interestingly, I don't have data here. Our previous paper also found some MSCs transformed into acinar cells in the CP pancreas. This suggesting that they may directly contribute to repair of damaged pancreatic tissue. This observation is promising for understanding how MSCs could support pancreatic healing. In the TMBS-induced mouse model, which displays features of both chronic pancreatitis and associated pain, we measured pain level using abdominal sensitivity. This was accessed using the Winefried filament test. This is a standard method for evaluating pain response in mice. In this test, a thin filament is applied to the mouse abdomen and the mouse response is observed. A positive response is when the mice raises, withdraws, or leaks its abdomen. This is a withdrawal reaction indicating pain. As shown in this figure, TMBS-treated mice responded to smaller amount of force compared to vehicle-treated controls, suggesting increased abdominal sensitivity and greater pain. However, mice that received MSC infusion showed a reduction in sensitivity to this stimuli, indicating that MSC may help lessen pain. So we believe that mast cells, which are involved in pain signaling, especially through the peripheral nerves connected to the pancreas, may play a role in the pain. Pain initiated from the pancreas also leads to a chain reaction of neuronal alteration all the way up to the dorsal root ganglia, or DRG, in the spinal cord. And DRGs are a cluster of nerve cells responsible for sending pain signals to the brain. That's why people complaining, I have my pancreas removed, I still feel the pain. Why? Because something has changed in the neuron system already. So MSCs may reduce pain by lowering inflammation in the pancreas and the DRGs. They could also potentially disrupt these pain pathways by influencing communications between the pancreas and the DRGs, a mechanism that we are actively studying in the lab. Encouraged by promising results from clinical animal studies, we are now evaluating the effect of MSCs in FDA-approved single-center phase 1 clinical trials for patients with chronic pancreatitis. These trials are funded by the National Institute of Diabetes and Digestive and Kidney Disease, also called NIDDK, and the Veteran Affairs Medical Center. For one of this trial, bone marrow will be collected from a healthy donor who passed all the necessary infectious disease and viral testings, and these cells were then carefully isolated and underwent rigorous quality control testing and potential testing to ensure they meet safety and potency standards. Once the patient is identified and agreed to participate to the trial, the MSC treatment will be proceed with the goal of getting positive results to support advancing to multi-center phase 2 clinical trial in the future. I want to mention here, like some other type of cells, of stem cells, unlike some other type of the stem cells, MSCs lose their effectiveness the longer they are cultured in the lab. To preserve their therapeutic potential, it's crucial to use MSCs at early passages for treatment. This is what we're doing here at MUSC. Now I'm introducing our first trial called the STEM-CAP trial. This is a study designed to test whether infusion of allogenic bone marrow-derived MSCs can help reduce pain in CP patients. We are going to enroll 48 CP patients for this trial. This is a randomized trial. Participants will be randomized either in the MSC group or the placebo group in a 1-to-1 ratio. The participant randomized to the placebo group will receive a solution resuspend the cells. The study will involve 5 inpatient visits over about 7 months. This visit will include a screening visit to determine eligibility, a baseline infusion visit, and 3 visit follow-up to monitor progress and outcomes. The major inclusion criteria are male and female between 18 and 75 years old, diagnosed with painful chronic pancreatitis for more than 6 months. They need to have a baseline ECBK pain score more than 30 points. They need to have stable dose of opioid for the past 30 days. Of course, patients with no opioid use are also welcome here. In addition, patients with acute pancreatitis and pain caused by other reasons other than CP, severe organ failure, and other issues listed here will be excluded from the trial. Potential participants are encouraged to discuss these special cases with our clinical investigators to find out if they're eligible. Our primary endpoint is to measure changes in pain levels from the beginning of the study until 6 months after. Our secondary endpoint will be a change in pancreatic volume, opioid use, quality of life, and severity of conditions. To ensure participants' safety throughout the study, we'll track all serious health events, monitor vital signs, and analyze lab results. The safety of the study will also be overseen by an independent data and safety monitoring board, our MUSC institutional review board, and the FDA. Additionally, we'll explore other measures such as changes in fecal elastase levels, endocrine function, to get a further impact on the MSC therapy. If you or someone you know might be interested in participating in this trial, please feel free to contact our wonderful clinical trial coordinator, Chloe and Leah. Their contact information are listed here. And you can also find more information about the study by scanning the QR code or visiting the clinicaltrials.gov website. We're also conducting a second clinical trial called MSC Pain Relief. This is a very similar study to the first one, but this one is specifically designed for veterans living with the CP pain. Unlike the previous trial I just introduced, this one will use each veteran's own autologous MSCs. We'll enroll 42 participants. And this is a crossover design, a little different than the other one. And all of the participants will receive a dose of MSCs either at the beginning of the study or at the 6 months after the study. Participants will be monitored for 18 months instead of just 6 months. So if you are a veteran with a chronic pancreatitis, we encourage you to learn more about this study. So when medical and surgical treatment are unsuccessful, some CP patients may undergo the total pancreectomy and islet autotransplantation. Based on animal studies suggesting that MSCs can protect the islet cells from dying after transplantation, we conducted our first clinical trial combining islet with patients' own MSCs for co-transplantation. We did 3 TPIT patients almost 8 years ago and published the data. The result of that trial showed that the procedure was safe. We are very lucky to get continuous funding from the NIDDK. We were able to expand this research into a larger clinical trial with 42 participants. In this study, 2/3 of the TPIT patients will receive their own MSCs along with islets, while 1/3 will receive only islet cells. We'll track participants for 1 year after their TPIT to evaluate change in islet function. So far, we have enrolled and treated, I think, more than 21, more than half of the participants. This is an incredible milestone made possible by an amazing team I'm lucky to work with. So if you are interested in cellular therapy for this type of study, especially with TPIT, please let us know as well. So none of this would have been possible without our incredible team. My long-term co-investigator, Dr. Charlie Strange, our dedicated physicians, lab scientists, clinical trial coordinators, and the GMP facility that made the cells. We are very grateful to our funding agency, the NIDDK and the VA. And most importantly, I want to thank you, the trial participant who trusted and believed in our work. I also want to help my son who is interested in research, who helped me prepare this talk. And thank you for listening.
Thank you so much, Dr. Won, for sharing your expertise and giving us a closer look at the world of stem cell therapies and the progress being made with these exciting clinical trials. We also want to express our gratitude to you and your team for your dedication to the pancreatitis community. Your hard work has brought these clinical trials to life, offering new hope to patients and families. We wish you continued success in this important work. Our second panelist, Alice Voss, who happens to be a registered nurse and a transplant outreach coordinator at Wake Forest Baptist Medical Center in North Carolina. But today, she's here with us as a patient with a history of chronic pancreatitis and share her experience of participating in a stem cell clinical trial for TPIAT patients at MUSC that Dr. Won referred to a few minutes ago. Alice, thank you for being here today. We are here and we are listening.
Oh, thank you for this opportunity that Mission Cure has given to me today to speak to you about my journey. My journey actually started in 2017 when I had my first occurrence of pancreatitis. And they decided that they would remove my gallbladder and thinking this would fix all my pancreatitis issues. And it did for about 5 years. But I was thinking I was home free. And then in 2022, I had a reoccurrence of pancreatitis. And the episodes of pancreatitis became more and more regular. For a year, I was being hospitalized about every 4 to 6 weeks with recurrent pancreatitis. My GI doctor ordered an abdominal CT scan, which showed I had some necrosis in my pancreas. I had genetic testing done, and that all came back negative. And then Dr. Jahan, my GI doctor, performed a procedure called an MRCP, where they went in and they removed the sphincter at the pancreatic duct. And unfortunately, I again had pancreatitis. And so I was diagnosed at that point with chronic idiopathic pancreatitis with some necrosis. I found myself becoming increasingly tired. And at the end of the day, I just had to go and rest. I had to go rest so I could get up for work the next day and do it all over again. And the weekends, I just wanted to sleep and catch up. And it wasn't until May of 2023 that my GI doctor informed me that we're just running out of options and that he recommended I go to a hospital that can perform a total pancreatectomy with islet cell transplant. And I'm just going to refer to that as a TPIAT. And I am thankful that Dr. Jahan, my GI doctor, was knowledgeable about this procedure because at that point he referred me to MUSC. So it didn't take long. And MUSC contacted me to schedule me for a first consulting visit about TPIAT. And at the same time, they contacted me about the clinical trial for stem cell and islet cell transplant. And after some thoughtful consideration, I decided to enroll in the clinic trial because this condition is not as well known or even understood as like other types of transplants, like kidney transplant or liver transplant or heart transplant. We've all heard of those, but who's ever heard of taking your complete pancreas out and then inserting those islet cells back into your liver? So I never heard of it. And so there my journey begins. So during my first trip to MUSC, I met with the study coordinator and Dr. Wang, and she's just awesome. They informed me that the trial would consist of 5 visits covering a span of 1 year. 3 of the visits lasted 2 to 3 hours, and the other visits were very short. They lasted about 30 minutes to 1 hour. I would go in and they would draw labs and do vital signs, and then I would fill out a questionnaire. During the long visits where it was 2 to 3 hours, that involved drinking a high glucose Ensure, and then they would monitor my glucose levels at the intervals of 15 and 30 minutes. I didn't feel like there were really any difficulties. I did have a challenge that I live in a different state. So I knew I had to travel to Charleston for all 5 of my visits. But all things considered, I felt like this was best for me to have the additional monitoring that the study provided. The recovery process was just a little longer than I expected. I felt like my strength and stamina for daily activities didn't return until about 6 months after my transplant. The recommendations from the surgical team and the study team were light exercise and walking. The most challenging part, I think, that you have after this whole procedure is that you are a diabetic and that you have to maintain taking Creon, the enzyme that breaks down food, for the rest of your life. We were hoping that the islet cells would completely take over, but I don't think that was my case. I think my case is that the islet cells are there to support me and keeps me from running extremely high. But for now, my life is pain-free and I have not been hospitalized in over a year. I do still manage my blood sugars with insulin and glucose, but by far, that is way better than the pre-surgery pain that I experienced when I had those multiple admissions. My biggest takeaway is that I am thankful for programs like this that give you extra support and provide realistic expectations for myself and my family members. This gives me the opportunity to hopefully help someone else in the future that will experience the same health issues. I would recommend that others that qualify for the clinical trial to participate. There are always 2 groups in a clinical trial. You have a control group and then you have your experimental group. The experimental group received the bone marrow biopsy and the control group did not. No matter which group you choose, which group you are chosen for, it will benefit you from the advancement of health science and research for pancreatitis. They were able to extract 688,000 viable cells from my pancreas, and I am just super happy about that. I would hope that the results of this study would help to educate patients and caregivers of the community on the outcomes of the TPIAT. Considering my experience both before and after transplant, I feel more educational information would be beneficial for all. I am thankful to have had the opportunity to participate in this clinical research of stem cells and islet cell transplant. I am excited to hear the results of this study for further advancement of our knowledge on effects stem cells may have on enhancing the production of islet cells. My hope is that patients might have improved outcomes in the future when diagnosed with pancreatitis. Thank you.
Thank you. And thank you so much to Alice and to Dr. Wang for joining us and sharing so much valuable information today. We have a lot of questions. One thing I do want to note is that we have a separate webinar on the total pancreatectomy and islet autotransplantation with Dr. Melina Bellin. That's on our YouTube channel and our website, so please check that out. Today, we are focusing on the stem cell therapy, and I'm going to start with a question submitted in advance. Dr. Wang, does stem cell therapy work, or should it work with patients with EPI?
So in animal models, we see, I showed some of the data that they improve pancreatic volume. So I expect they are, but nothing has been known unless we complete the trial and compare, analyze patient data. So far, I don't know.
And have umbilical cord MSCs been tested in clinical trials? And aren't they more potent than bone marrow or other sources?
Yeah, we also ran a clinical trial using umbilical cord derived cells for our type 1 diabetes patient with new onset type 1 diabetes. We do believe that there are subtle difference between bone marrow derived MSCs and umbilical cord derived MSCs, but it's really hard to tell and unless you compare side by side in similar clinical trials, which is almost impossible in human. But we did in the past years, we did do a lot of molecular and cellular analysis, compare the differences. So far, there's no much conclusion. I think most people believe that because the umbilical cord MSCs are from baby, they're not from the mother, they're from the baby. So they are probably younger than the bone marrow MSCs. Because remember, I mentioned that the ability of bone marrow derived MSCs to cure decrease as we grow cells longer. So, there's a general concept that, if you get bone marrow cells from older person, they are not as potent as the younger person. If we understand that from that perspective, umbilical cord derived cells are younger than the bone marrow MSCs. But we also, however, with this being said, sometimes the bone marrow derived MSCs has more immunosuppressive function, depends how you evaluate it. I think they both have their pros and cons. They're indeed, so altogether, there are indeed some subtle differences.
Thank you. And there is somebody here who has stored their child, who is now a teenager's cord blood since birth. They want to know, could those be used in the clinical trial?
Not for our trial. Our trial, we already have the cells from a very healthy donor. We currently frozen these cells in the liquid nitrogen. We have to use these cells. This is the study approved by the FDA. We don't have the permission to use patients' own cells at the time being.
And then I think you touched on this, but somebody asked, why did you decide to use these allogenic, like cells taken from other people rather than cells taken from the patient themselves?
Yeah, like I said, the first reason you have to know if the disease impact their so their allogenic and autologous, they both have their pros and cons. If you use patients' own stem cells, the good thing is there's no immune rejection, you get it from your own. However, you have to go through the collection process. I mean, if you do, this is not easy for blinding also. Blinding because you don't want to tell the patient what they get, because the placebo effect for pain can be more than 60%. It's a very high placebo effect. The beauty of use allogenic, another people's cell, you can frozen there, you can thaw them whenever you have patient identified. You don't have to wait to grow the patient's own cells. But there is a chance to be rejected by the body. But this is not a very dramatic issue for MSCs because they're immunoprivileged. So that's why you see I presented two studies. One used the allogenic cells, the other used autologous. So, so I think at the time being, both can be used, and depends on the study design, they're using different type of cells.
If I receive stem cell therapy for my knee, and you did mention this in your presentation, would it also target my pancreas, or would that have to be a completely different stem cell procedure?
That's a great question. So for the knee therapy, I believe most of the people that do the local injection. So maybe they're more realistic to, more concentrated in the knee area. However, if you give MSCs intravenously, which is most clinical trials do, they probably able to migrate to different tissues, organs, and benefit from different diseases. I think we said that MSCs are very rare adult stem cells in the body. Their tendency is to migrate to injured areas. I think they will have beneficial effect for other as well. However, if you inject locally to the knee, maybe it's more beneficial to the knee other than the pancreas.
Right. Can stem cell therapy be used with chronic necrotizing pancreatitis?
I wish to tell, I have the answer, but this is a great question. I don't really know, unless we, conduct a well designed study, we won't know the answer. Sorry.
Is this trial only for Americans, or will UK and European candidates be considered?
We don't define a patient based on where they are, but we do have, you do have to have multiple visits for each trial to make it possible. I think anybody can participate, but you have to make sure you're able to conduct the in-person visits.
Is there an increased risk of cancer from using MSCs because of cellular change and proliferation?
Yeah, that's a very big, valid concern. And in one of the slides, I didn't go to details. There are a couple, of review papers describing that. So in a 2021 review paper, this group reviewed MSC therapy in the past 15 years. I think they evaluated data from 62 clinical trials, enrolled about 3,500 patients for almost 20 different diseases. And they didn't show any evidence of, cancer development in that study. That's as far as I know. We haven't heard of any evidence in human yet.
This is fantastic, and we have more questions coming here. I want to insert one of my own questions here that I did not give you a heads up on, which is everybody here can tell that Dr. Wang is like one of the world experts, most respected in stem cells research and therapy. But there's a lot of stem cell therapy being marketed in various countries around the world. And can you comment on kind of the quality of what people might find if they are see an ad for a stem cell therapy somewhere in another country outside the US?
Yeah, so US, I think in the US, FDA is considering give a license for MSC. I think it's the company's Mesoblast. They're going to may get the approval for BLA biological license in the new year for acute refractory graft-versus-host disease. And MSC has been approved for certain diseases in a couple of countries, but not all countries. If I saw an advertisement showing MSCs can cure this and that, I'll be very careful. The reason is, if it doesn't hurt you, fine. But the quality of the cells need to be very much guaranteed. All our study is monitored by the FDA. We have doesn't call license, it's called investigational new drug approval. Each study is under very FDA approved protocol. According to this protocol, our facility agree to grow the cells for example, most of the time 21 days. And during these 21 days, we have to do a series of very stringent testing to make sure the cells are at sterile, no contamination of mycoplasma, bacteria, or any endotoxin. And also these cells need to pass the what's it called? Pass the potency test, meaning that if we use in vitro experiment to predict the function, although you can never be accurate. I mean 100% accurate. So these cells are very they produce it's called good manufacturer practice standard. You don't have to worry about contamination and things like that. So I think to answer your question, I'll be very careful selecting those treatments, especially not something that are not approved by the FDA.
Thank you. There's a lovely note here from Anne, which Dr. Wang can read later, but her question is, could this be applicable to autoimmune pancreatitis as well? Or would the autoimmune cells attack the MSCs?
Yeah, so far we believe that MSCs are immunoprivileged. They are not going to be destroyed by the immune system. But it's hard to know after they are injected into the patient. So good thing is even they are destroyed by the immune system, they already, their studies showing they already changed the macrophage that are important for some disease. And also they secrete very small tiny particles. Remember, I was too fast in my presentation. Very small particles called extracellular vesicles, mitochondria. Those are the mediators that can mediate their function. So I think I'm not worried about that too much. That's but it's hard to know after injection.
How many years would it take if this phase 1 study is successful to for this type of therapy to be available more widely to patients in clinical practice?
So this study is for 4 years, and we plan to enroll 42 patients. We already, I think, have 2 participants for STEMCAP1. And we are supposed to finish in 4 years. And I think there's a long way to go to clinic because we have to do a phase 2, phase 3 clinical trial if the data is positive. So I think it takes a while.
Recently, there was news of a Chinese experiment where a diabetes patient was injected with stem cell-derived islet cells from her own fat cells, and now she does not need insulin. Is there a study that will explore this, especially for people who've had TPIAT?
Yeah, that's a great question as well, because we wanted to do that for a while. So the reason is that you make, so basically that's use a different type of cells. I think it's use inducible pluripotent stem cells. Remember the second category of stem cells. So my lab studied that as well. We have a Dr. Wei, he differentiate inducible pluripotent stem cell into beta cells. And that's basically targeting type 1 diabetes. And Vertex has already done a lot of patients in type 1 diabetes with exciting results. However, if we apply this to chronic pancreatitis patients, specifically to those who have, undergo total pancreatic islet transplantation, they don't have enough islet cells. That's, if you give those iPSC-derived beta cells, there would be no immune rejection. And that's my hope to get funding to start trying. It's on our, we plan to apply for grants, but from the thoughts to the funding to able to do it, it takes a while.
Okay, and one more question, two-part question. One person asked, would this help with genetic chronic pancreatitis? And the other person asked, would this help with idiopathic chronic pancreatitis?
That's a great question. So, so basically, to answer that question, if we complete the trial, we can divide the patient into subcategory and we see which one responded better. But that's need a lot of data to answer the question. But that's a great one. Thank you.
Wonderful. Thank you, everybody, for submitting your questions. If we didn't get to you, we will try to do that by email, but I think we got most of them. And thank you so much, Dr. Wang, for sharing your expertise with us.
Thank you for the opportunity.